Hey everybody, welcome back to another video. And in this video, we're going to start talking about lipid digestion and absorption. And in the next video, we're going to continue filling out this diagram when we talk about lipoprotein metabolism. Okay, but for now, let's focus on the digestion and absorption of lipids. Now, when we talk about this, we have to talk about what are the most common dietary lipids that we're consuming. Well, they come in the form of triacylglycerols, which we've talked about before. They come in the form of phospholipids, which again we've talked about before. They also come in the form of cholesterol and cholesterol esters. Okay, now you guys know what cholesterol looks like, but a cholesterol ester is essentially just a cholesterol molecule, but with a fatty acid attached to the end of it instead of that hydroxyl group, okay? So that's essentially what a cholesterol ester is. And some foods will have cholesterol ester. For example, an egg yolk contains about 10% of its cholesterol is in the form of cholesterol esters, okay? Some foods have more, some foods have less. But the entire thing is that in your diet, right, these are the kind of lipids you're getting. Your triacylglycerols, your phospholipids, your cholesterol esters, and then those plant sterols that we talked about beforehand called phytosterols, okay? Phytosterols. All right, so these are the kind of lipids that we're going to be dealing with, and we're going to see how they're going to be digested, aka broken down, and then how are they going to be absorbed again at the level of the intestine. So there's a couple of places where digestion takes place, okay? One is at the oral cavity and another is at the stomach, but we're going to combine these two because they have a very similar enzyme. And then the other one is at the level of the duodenum, okay? Well, really at the level of the small intestine, but we'll say that it's at the level of the duodenum and maybe a little bit in the jejunum, okay? Okay, so first things first, let's go ahead and take a look at the digestion of a triacylglycerol first, okay, or a triglyceride. Because a triglyceride starts its absorption at the oral cavity and the stomach, okay? But really, these two combined, the oral cavity and the stomach, they don't contribute to a majority or a very major digestion of a triacylglyceride, primarily because food doesn't, stay in the oral cavity for a long period of time, and the enzymes that are performing this are not very efficient at their job. And we'll talk about that a little bit later why. But this molecule right here that you're looking at is a triglyceride, okay? We've seen this structure before. It's a triglyceride. And you can see that, again, it has that SN1 carbon, SN2 carbon, and SN3. If you guys are unfamiliar, SN1 is stereospecific number 1, 2, and 3, okay? And the oral cavity produces a specific enzyme by the sublingual salivary glands, okay? And that enzyme is called lingual lipase, okay? So the sublingual gland makes that, and it's called lingual lipase. So first enzyme we're going to have to be dealing with is this guy right here called lingual lipase. But there's another one. There's another one. Since we're combining the oral cavity and the stomach together, we're going to say that the stomach, from the chief cells of the stomach, if you guys remember the chief cells, they also make an enzyme called gastric lipase, okay? Now, lingual and gastric lipase, they both are cleaving or functioning at an exact same space, okay? And if you guys remember, the whole goal of this digestion process of a triglyceride is to gain some free fatty acids, okay? And you guys remember that there was a bond, a special bond that attached the glycerol molecule to the actual fatty acid, okay? And that bond, that special bond, was actually this guy right here. It was called an ester bond, guys. It was called an ester bond, okay? If you need a reminder, here's how an ester bond actually looks like. Right, so here's a carbon, doubly bounded, R, oxygen, and another R, okay? So this is how an ester actually looks like, okay? This is what an ester bond is. Here's one hydrocarbon, here's another hydrocarbon, okay? All right, or again, this ester bond. So this lingual lipase, okay, let's show it as a kind of scissor, Okay, it's kind of, let's show it as a scissor. Okay, because it is an enzyme after all. It'll come here and it'll cleave that. Once it cleaves this bond, okay, this ester bond right there, specifically at the SN3 position, okay, we call that process hydrolysis, guys. Okay, we call it process hydrolysis, okay, and in that process, if you remember, we talked about this. We said that we're actually adding in water in the reaction. That's why it's called hydrolysis. Okay, now. Something very cool about this lingual lipase and gastric lipase is that they both act on the triacylglycerides that contain medium chain fatty acids or short chain fatty acids. Okay, so they act on the triacylglycerols that contain medium chain or short chain fatty acids. Okay, and they can actually work at low pHs, okay? And that's, for some of you wondering, gastric lipase, how does it work in that low pH? Well, it's designed to work at a low pH of the stomach, okay? Because remember, gastric lipase is secreted by the chief cells, okay? And that's in the inside of the stomach. And in the stomach, we're also making that hydrochloric acid. So that's just some facts about the enzymes, okay? All right, okay, so once we actually hydrolyze this triacylglyceride, which we said probably contains short chain or medium chain fatty acids, what you're going to end up getting is this molecule right here. Okay, so you have fatty acids attached to the first carbon and the second carbon, but no fatty acids attached on the third carbon. For that reason alone, we actually end up calling this guy a 1,2 diacylglyceride. Okay, why 1,2? Because there is a fatty acid on the 1 and the 2 carbon, and there's only 2 fatty acyls, so we call it a diacylglycerol, okay, because that tri is gone. So we formed a diacylglycerol from this hydrolysis, but we also free something else. We freed either a short chain fatty acid or a medium chain fatty acid, depending on what was actually esterified here, okay? But really, we didn't actually cleave the majority of the triacylglycerides in the diet right there. So we actually left a remainder of 70%. 70% of the triglycerides actually remain and go off all the way to the small intestine, which means what? Guys? Which means that this lingual and gastric lipase together contributed to about 10 to 30% of the triacylglycerol digestion. Okay? So that's interesting. That's interesting right there. Now, with these guys moving on towards the duodenum, okay, now we can get to look at how the actual triacylglycerol is going to be absorbed and how the cholesterol and the phospholipids are going to be absorbed. But before we take a look at the triacylglycerols, let's take a break real quick, okay? So remember I said, and let's do it in this way over here. This triacylglycerol, okay, it'll come in, and it'll be in this shape right here, okay? Why do I have it in this kind of blob format? Why do I have it in that blob format? Okay, I'll tell you why I have it in that blob format in 1 second. But when you guys are actually ingesting fats, okay, these fats are going to kind of aggregate with one another and form this giant droplet. Okay, they're gonna form something known as a fat globule or a fat droplet. Okay, and that fat droplet is consisting of those triacylglycerols that we just had, okay, the fatty acids, and the 1,2 diacylglycerol. Okay, so we can write that down, the 1,2 diacylglycerol and the free fatty acids. Awesome, guys. Awesome. Now, let's remove this stuff. Let's get rid of all of this. So here's that fat droplet that is arriving from the actual oral cavity. okay, after the digestion, after hydrolysis. That guys are now arriving towards the duodenum. What other things are arriving with it? Well, the other things that are arriving with it are going to be free cholesterol from the diet, and cholesterol esters, and a phospholipid. What kind of phospholipid? Well, this specific phospholipid we've actually talked about before. It's called a phosphatidyl choline. And if you guys remember from the previous video, we called it a specific name. We said that it was actually going to be called lecithin. Okay? Lecithin. So this right here is a phospholipid that is called lecithin. And this right here is the actual choline group. Okay? Or sorry, right here is the choline group. Okay. Now, what specific enzymes are going to act on these guys? Well, something else I got to mention is that when we're at the level of the duodenum, okay, these enzymes that we're going to use are actually not made inside of the intestine. They're coming from somewhere else. They're actually coming from the pancreas, guys. They're coming from the pancreas. And if you guys remember, we've repeatedly said this in many videos, but the pancreatic duct and this actual gallbladder, okay, will fuse together to make one giant duct, okay? And that duct will then open up into the small intestine, or the duodenum, okay? It's the hepatopancreatic ampulla. And that hepatopancreatic ampulla is surrounded by the smooth muscle, okay, which is a sphincter. It's called a sphincter of Oddi. And in the presence of a hormone called cholecystokinin, this sphincter will actually relax and open up and release the digestive enzymes and bile contents. So what is it releasing, guys? It's releasing many things. It's releasing, and you know what? Let's color code these. Let's color code these. So let's keep consistent with the enzyme color. This is the enzyme color. It's releasing a specific couple of enzymes. The first one is going to be called PLA2, or also called phospholipase A2. The other one it's releasing is called cholesterol esterase. Okay, cholesterol esterase. It's also releasing this special molecule that we'll talk a little bit more about. It's called colipase. Okay, and these are all coming from the pancreas. Another enzyme that's coming in is called, and this is crucial in lipid digestion, it's called pancreatic lipase. Okay, and finally, something else that's extremely crucial, guys, that's coming out of this is going to be, let's make it a little bit of a darker color. It's called your bile, okay, and specifically your bile salts. And I'm not going to mention them too much because we've already talked about them in other videos. Go and watch those. But the bile salts are coming out. Now, you guys know that that's not everything that was in the bile, okay? You guys know that the bile had actual cholesterol and phospholipids in it as well, okay? So I'm going to put this out here, but the bile also had cholesterol, again, free cholesterol, and it also had phospholipids, okay? So PL for phospholipids. Now, the cholesterol and the phospholipids that is released from the actual bile into this small intestine right now, that will also go through this digestion process that we're about to talk about, okay? That's why I brought that up. But another reason why I brought this up was because these are the enzymes that we're going to use in this small intestine to actually go ahead and start digesting a lot of these molecules. So first, let's talk about the cholesterol ester. So this guy right here is a cholesterol ester. One thing you have to know is that free cholesterol does not require any digestion. It'll get readily absorbed. So this cholesterol from the bile and the cholesterol from the diet that is free, meaning it's not bound to any free fatty acids or esterified, those will get freely absorbed. Remember that. But if it's in a form of a cholesterol ester from the diet, guess what? It needs a specific enzyme. And that enzyme, guys, is going to be called a cholesterol esterase. Okay, so it'll come over here, that cholesterol esterase, and it'll actually, and let's write it down. So here's cholesterol esterase. All it is going to do is it'll cut that free fatty acid, okay? that's bound or esterified to this actual cholesterol. And what you're going to get out of that reaction is going to be two things. What you're going to get out of that reaction, again, let's keep consistent with the colors, okay? Just to really make it pretty for you guys, is going to be a free fatty acid and a cholesterol molecule, okay? So you're going to get a cholesterol and a free fatty acid, okay? Awesome. Okay. Now, the phosphatidylcholine or the lecithin, I said that was also coming out of the bile. So that guy will also get digested in the way that I'm about to talk about right now. But there's also this phosphatidylcholine in the diet, okay? Now, the enzyme that is actually going to be working here is going to be called phospholipase A2, or that PLA2. And it's going to specifically cut at this SN2 or second carbon position. Second carbon position, and right here is going to be it. Okay, here's that guy right there. It's called phospholipase A2. And this guy, from the product of this guy's digestion, we're going to get two things. Two things. So first, we're going to get a free fatty acid. And because this is a phospholipid and it's on the second carbon, it's most likely going to be an unsaturated fatty acid, as you can see here. And the other thing that we're going to get is actually a product called lysolecithin, which, if I were to draw it out very quickly for you guys, would look something like this. So here's what it will look like. This product right here is called the lysolecithin, and let's write that down like this. Here's a lysolecithin. And it's important to understand the name here, okay? So the original molecule was a phospholipid called lecithin, okay? But then I went through the process of digestion and I lysed it, I cut it. That's why it's now called lysolecithin, because it has been cut, okay? And it's specifically been cut at that second carbon position and gave us a fatty acid and this lysolecithin. Okay, now, what about the triacylglycerol? Okay, so now we talked about the phospholipids and the cholesterol being digested. What about those triacylglycerols and the remainder of these guys? What about that big fat droplet? Well, the big fat droplet is too hydrophobic to interact with the chyme. You guys remember how from the stomach, okay, we actually created chyme. And that chyme, remember, it had some hydrochloric acid and it had some water in it, okay? And that guy was pushed out through the pyloric sphincter into the actual duodenum, okay? And once it was in the duodenum, you guys remember that this pancreas started secreting all these enzymes, okay? And these bile salts. And at the same time, it would also secrete some bicarbonate, which would neutralize the acidity of the chyme from the stomach. Okay, and this is important because it's going to allow for all of these enzymes to work properly. But something I didn't actually tell you is that these enzymes are secreted in the watery portion of the actual duodenum, okay? So right here is going to be the actual watery secretions, or the actual chyme, okay? And inside there is going to be the enzymes, such as the pancreatic lipase, for example, that will help digest this big fat globule. But this is the problem. This fat globule is hydrophobic, right? And this is mostly water. So we got a problem. This big fat globule that's hydrophobic doesn't want to interact with this water. It sits on top of the layer. It actually floats on top of it. So what do we need to do? We need to break this guy down into smaller bits and pieces so it can easily interact with the water layer and sink down deeper and deeper and deeper and interact with those enzymes. How do we do that? How do we do that? Well, we use this special guy right here called the bile salts specifically. So these bile salts, guys, they're going to start coating the actual big fat droplet here, okay? And they're going to emulsify this guy. They're going to emulsify it. So here's the process of emulsification. And once this dude gets emulsified, look at what's going to happen to it. Look at what's going to happen to it. It'll break down into smaller particles. So here's a smaller fat droplet, okay, smaller fat droplet. And it still has that actual the bile salts surrounding it, okay, stabilizing it, if you will. Now, here's something about the bile salts, okay? It does help emulsify the actual big fat droplet into these smaller little droplets, okay? But there's another problem. The bile salt is actually inhibiting the pancreatic lipase. What do I mean by this? I mean that the pancreatic lipase is supposed to come and bind to this big fat droplet and start breaking down the triacylglycerols and the 1,2 diacylglycerols in there. But the bile salt that's actually surrounding it is preventing it from happening, okay? It basically displaces the enzyme from the substrate at the surface of the lipid droplet, and we can't do anything there. So in order to actually solve this problem, the pancreas has already thought of a solution. It secretes another enzyme here called colipase, okay? And colipase possesses hydrophobic regions. Now, bear with me here. This fat droplet is hydrophobic, okay? And so if the colipase possesses a hydrophobic region, it means that it can associate very strongly and bind to this actual fat droplet, okay? And another thing is that it also has the ability to bind to the pancreatic lipase. So let me show you something. And let me depict it in this way so you guys can fully understand what I'm trying to say. So this guy will bind, okay? This guy will bind to the actual fat droplet, okay? Because it has specific hydrophobic residues. And it allows now for the actual pancreatic lipase enzyme, which we'll do in this color, to bind here, okay? So let's do it like, let's do it like this. Boom and boom. Oh my God. Okay, so there you go. So this right here in purple is the pancreatic lipase, and in green right there is the colipase. Now, here's what happened. The bile salts inhibit the pancreatic lipase from binding to the actual fat droplet, okay? So what ends up happening is the colipase binds first to the actual bile salt stabilized fat droplet, which then allows the pancreatic lipase to bind to the fat droplet. Okay, now the pancreatic lipase can initiate its job and start hydrolyzing all these triglycerides and 1,2 diacylglycerols. Okay, and if you guys are even more curious, this process also requires, for example, calcium as well. This process also requires calcium to be present. But neither here nor there. The pancreatic lipase, where does it cleave? Where does it like to cleave? It likes to cleave primarily at the SN1 position. Okay, it likes to primarily cleave at this position. Okay, so if I go back, here's the SN1 position, meaning that the pancreatic lipase would like to cleave this part of the triacylglycerol. But the second position that it likes to cleave is the SN3 position. Okay, so it firstly cleaves at the SN1, but it also has some affinity for the SN3 position as well. Okay, and again, on top of the calcium and the colipase, it also requires a high pH to work, which is why we secreted the bicarbonate from the actual the pancreas. Okay. All right. So one more time, because I know this is very challenging. We had this fat droplet, which is hydrophobic. Okay. And the problem was that it can't actually go and interact with the water layer of the chyme inside of the duodenum. So what do? We bring in the bile. The bile will come and bind to this guy and emulsify it into these smaller little fatty droplets. Okay. Now, this guy can actually sink down and interact with the enzymes that were secreted in this actual water layer. But here's the problem. The bile on the surface of this actual fat inhibits the pancreatic lipase from working by displacing it from the actual substrate site. So the pancreas thought of another solution. at the same time it secreted all of this, it secreted colipase, which has a hydrophobic residue that interacts with the actual hydrophobic fat molecule, and it also associates very strongly with the pancreatic lipase. So it kind of serves as like a docking site or like a linking site for the pancreatic lipase to bind to and allow for the cleavage of the triacylglycerols at the SN1 and the SN3 position, but primarily at the SN1 position. Okay. Now, with all this said, what are the products of this digestion? What am I going to get? Well, I'm not going to fully show it to you here, okay? But if you want, you can actually draw out a triacylglycerol for yourself and cut them at the SN1 and cut them at the SN3 position, and you will see that what we will end up with is going to be a 2-monoacylglycerol and some long-chain fatty acids, okay? And also, we might get something called a 1-monoacylglycerol, which is a result of isomerization by the 2-monoacylglycerol, but you don't have to worry about that too much, okay? All right, so, man, this is a lot of things getting digested here, okay? And those are the digestion products are in blue. Okay, but guess what? We're not done yet. We're not done yet. We actually have to digest this guy even further. And guess what? When the pancreatic lipase digests these fat droplets, this fat droplet will keep shrinking in size and size and size until it forms this very small, negatively charged aggregate of lipid molecules. So here it is, right here, and let's say there's one here, and there's another one here, okay? And they're still sort of lined by the actual bile, okay? Right there, and right there, okay? So these guys are negatively charged aggregates of lipid molecules. Aggregates just means that they're kind of just smushed together all around, okay? And these guys are actually called micelles, okay? Micelles. These are called micelles. Negatively charged aggregates of lipid molecules. And just for your understanding, this guy right here was about 50 to 100 nanometers in diameter, whereas the micelles are about 5 nanometers in diameter, okay? So this is just to put into perspective how much smaller this guy actually got, okay? Now, these micelles, they're small enough to actually fit in between the actual brush border of the enterocytes, okay? But here's another thing. What's actually inside of these micelles? So I just told you that they're positively charged aggregates of lipid molecules, but what the hell is actually in them? Well, everything that we just talked about is in them. So let's kind of show it right here. So the free fatty acids, okay? The 2-monoacylglycerol, the long-chain fatty acids, and the 1-monoacylglycerol, the cholesterol, this other free fatty acid, the lysolecithin, okay? All of these guys are inside of this actual micelle. They're all inside of the micelle. And you know what else is inside of the micelle? Your vitamin A, D, E, and K, your fat-soluble vitamins, okay? These are all inside of the micelle as well. All right? All right, awesome. So your fat-soluble vitamins will also hop on a ride inside of this micelle. Why? Because they're fat-soluble. Okay, so now let's take a deeper look. Let's actually zoom in on this interaction, and let's take a look at what is happening here. So here's going to be our micelle, okay? And inside of it, we had a couple of things, guys. We had our lysolecithin. I'll just put lyso. We had our monoacylglycerol, okay? The two monoacylglycerol, the 1-monoacylglycerol. We had our long-chain fatty acid in there. What else did we have, guys? We had some cholesterol, okay? And I didn't show any digestion here, but we did have a phytosterol, okay? Remember how I said one of the common dietary lipids is a phytosterol, which is just really a plant sterol. You can think of it as a plant cholesterol, basically. And it also uses the same exact digestion mechanism as the cholesterol ester, basically, okay? So maybe we will have some phytosterol in there, Okay, and finally, we'll also have some vitamin A, D, E, and K all inside of this micelle. Now, when this guy gets close enough, okay, the actual bile acids or the bile salts, okay, that were actually stabilizing this guy say, all right, I'm out. The bile acids will go out. And they will actually circulate, well, not circulate, but they will keep carrying on within the GI tract all the way to the level of the distal ileum, okay? And at the distal ileum, let's show it like this. At the distal ileum, about 97% of this actual bile is going to get reabsorbed, okay? And again, I'll write that for you guys at the distal ileum, okay? And it'll get reabsorbed in an energy requiring process, okay? Energy required in this absorption process. Again, it'll go 97% throughout the hepatic portal vein, and it'll go back to the liver where it came from. And this right here, by the way, is called the enterohepatic circulation, okay? Or the enterohepatic cycle. Okay, so one more time, as the micelle gets close here to the actual enterocyte, the brush border, the bile salt says, I'm out, right? So you don't see any green around it. And the bile salt was actually stabilizing this guy, okay? And without the bile salt, what ends up happening is this, all the contents of this guy will start to disappear, okay? Well, not disappear, dissipate. They will start going out and getting absorbed at the level of this enterocyte here. Actually, where are we, by the way? Where are we right now? So right now, we are actually at the level of the distal duodenum and jejunum. Okay, so when it comes to the absorption, we're actually at the level of the distal duodenum and the jejunum. All right, now, what does actually the absorption look like? And one more time, I really do want to mention that these micelles don't actually cross this actual plasma membrane of the enterocyte. They're basically only there as a temporary structure to deliver the digested lipids to the enterocyte, okay? And then they kind of break down and release their contents, okay? So with that being said, for example, you can have two types of fat digestion. You have two types of fat digestion. And specifically, when it comes to lysolecithin, monoacylglycerols, and fatty acids, okay, you actually have two types of absorption, okay, so two. One of them is actually protein dependent, okay, it's a protein dependent model. The other one is actually a protein independent model, okay, the protein independent model. And in the protein independent model, it's actually a diffusion model, okay, where things just passively diffuse across the actual membrane. All right. And that basically just is your regular concentration gradient. Okay. So if the concentration of, for example, of monoacylglycerol is higher here than it is inside of the enterocyte, then this guy will just passively diffuse along the actual membrane. Now, there is some thought that maybe you have to remodel certain phospholipids inside of the membrane. For example, shifting fatty acids between the first and second carbon positions in some phospholipids. But again, you don't have to worry about that. That's too in depth. I just want you to know that there's a protein independent model, which is a diffusion model, okay, down the concentration gradient, all right, and there's a protein dependent model. I'm not going to talk about the protein independent model because it's pretty simple. It's just diffusion, okay, high amounts here, low amounts here, boom, you diffuse. What about the protein dependent model though? And again, remember, these two are only for the lysolecithin, the monoacylglycerol, and the fatty acids. The protein-dependent model is a little different, okay? And it requires a couple of different transporters, okay? So this one, for example, CD36, is a fatty acid transporter, but it can also transport monoacylglycerols. So let's take a look. Long-chain fatty acid right here, and a monoacylglycerol. Both of these guys use the same transporter, and voila, you actually get the long-chain fatty acid coming in. Okay. What else? What else? Well, with the lysolecithin, there is actually, we don't know if there's a protein transporter or not. Okay. There is actually a protein transporter identified in the yeast, but we don't know if it actually exists in humans. So we can assume that it goes through an independent protein independent pathway. And here it is, the lysolecithin coming in here. Okay. And just for the sake of space, we can show a monoacylglycerol here as well. There is also this other protein here called fatty acid transporter protein 1,4. Okay. This one is a little different. It's not actually a transport protein. So CD36 is a transport protein. It takes a fatty acid and a monoacylglycerol and brings them into the cell. But this guy's actually not a transport protein. So what is it? Well, it actually takes free fatty acids that are already inside of the cell, and it kind of acts like an enzyme. It adds something called a coenzyme A to it, okay, and creates something called a fatty acid CoA. The skinny on it is basically that by creating this fatty acid CoA, it's priming the fatty acid for being turned into either a triacylglycerol, a phospholipid, or a cholesterol molecule. But how does this fat P1 by doing this increase the absorption of fats? I'll tell you how. Because it's changing the concentration gradient. So by decreasing the amount of free fatty acids that's available inside of this actual enterocyte, by turning it into this fatty acid CoA, it maintains a steady concentration gradient for any fatty acids that are out here to be able to come in here, either via protein independent or protein dependent model. Okay, so that's how this fat P1 is actually facilitating greater absorption. Okay, now that's how those guys are getting in. What about the actual cholesterol? Okay. Oh, and one more thing, actually. One more thing is that when these guys actually come into the actual cell, again, the cell is basically filled with a whole bunch of water, so these guys need carrier proteins. So they actually have these things attached to them when they come in, and let's put it right here. So lyso, they have these things attached to them called and let's do it like this. They're called fatty acid binding proteins, okay, that will just deliver them towards the endoplasmic reticulum. And I'll talk about why they're going there, okay? And same thing with the actual monoacylglycerol. They have the fatty acid binding protein to it. And same thing with the actual long-chain fatty acid, okay? Now, what about that cholesterol? The cholesterol and the phytosterols, they actually use the exact same transporter. They use the exact same transporter, and that transporter is called Niemann-Pick cholesterol 1L. Okay, so or you can also just write it as NPC1L1. Okay, and this guy is going to take your cholesterol. Okay, your cholesterol, and it's going to take your phytosterol. All right, and it will bring both of these guys in. It'll absorb both of these guys, okay? And what you'll end up getting is going to be a cholesterol and a phytosterol. But you know something funny is that the phytosterol that comes in, almost none of it will actually become absorbed because of the presence of these two proteins that are adjacent to the NPC1L1. These are called ATP binding cassette globulins 5 and 8, okay, or ABCG5 and 8. These guys will immediately take the phytosterol that came in and boom, push it back out inside of the lumen or the small intestine so that it goes and gets excreted. Additionally, this guy can actually block cholesterol absorption. This phytosterol, it can block cholesterol absorption through many different mechanisms. It can displace it from actually being inside of the micelle. It can compete with cholesterol by binding to the NPC1L1 transporter, and yeah. Additionally, some of the cholesterol can actually use this ABCG as well, and it can come out here. And for that reason, because some of it will get pushed out rather than absorbed, the overall efficacy of cholesterol absorption is about 50 to 60%. Okay, so about only 50 to 60% of the cholesterol is actually going to be absorbed, and that's again because some of that cholesterol will travel back using the ABCG transporter and get pushed out into the actual lumen. Now, once the cholesterol actually comes in, it will also bind to a special protein, and that's called the sterol binding protein. Okay, why are they binding to all these proteins? Why are the fatty acids, the MAG, the lysol, the cholesterol, why are they binding to these proteins? Well, because they're fats, okay, and fats are hydrophobic, and inside of the cell is pretty much a lot of water. So they're binding to these proteins so they can be safely carried to the endoplasmic reticulum. Why are they being carried to the endoplasmic reticulum? Well, here's something funny. We spent the entirety of digestion breaking these guys down to their small constituents and what they're made up of. But now, inside of the endoplasmic reticulum, we're going to reform them again, okay? And this is pretty hilarious. So watch. Here's what we're going to do. We're going to grab, and let's do it with this red color, okay? We're going to grab a lysolecithin and a let's write it here, and a long chain fatty acid, okay? We're going to grab those two together, all right? And we're going to make a phospholipid, okay? Additionally, we are going to grab a monoacylglycerol, okay? So we're going to grab a monoacylglycerol and another long chain fatty acid. And with that, we're also going to make a triacylglycerol, okay? Because we're taking a monoacyl and we're adding a couple of long chain fatty acids to it, precisely 2, and we're going to make a triacylglycerol, okay? Okay, what else do we need to resynthesize? Well, with the cholesterol, we're actually going to take that cholesterol, okay? And we're going to grab the actual long chain fatty acid here, and with that, we are going to make something called a cholesterol ester. We're going to make a cholesterol ester, okay? So you see how we just resynthesized every single thing that we spent the whole time breaking down? Yeah, we just did that, okay? Now, there are specific enzymes that play a role here, okay? And I'll just briefly mention them. So the enzyme that's actually working to create the phospholipids is called an acyltransferase enzyme, okay? The enzyme that's taking the triacylglycerol, that's making the triacylglycerol, it's called an acyl-CoA acyltransferase. Or actually, sorry about that. It's actually going to be an acyl-CoA synthetase. Acyl-CoA synthetase enzyme, okay? And then finally, the enzyme or enzymes that are going to be taking the cholesterol and combining with the long chain fatty acid to make a cholesterol ester are going to be called a ACAT1 and ACAT2. So ACAT 1 and 2, and ACAT basically just stands for acyl-CoA cholesterol acyltransferases, okay? All right, and again, they are esterifying this actual cholesterol, okay? Not the plant sterol, okay? 1, because they just can't do that, and 2, because the plant sterol doesn't even make it in here. It makes it in and it's immediately pumped out, so none of it is actually absorbed. Okay. Now, why did we have to bring these guys in the ER? Well, we brought them into the endoplasmic reticulum because we wanted to resynthesize them, but more importantly, now we actually have to find a way to transport these fats, okay, which are, by the way, not soluble in water, which means they're not going to be soluble in the blood. We're going to find a way to transport these fats in the blood. So what do we do here? Over here, we're going to combine these phospholipids, these triacylglycerols, these cholesterol esters, and perhaps some of the fat-soluble vitamins as well. Fat-soluble vitamins, okay, in here. And we are going to make a special structure, okay, that we'll talk about a bit more later in the next video. Actually, a lot more later in the next video. But this structure is going to be called a chylomicron, okay? It's going to be called a chylomicron, and this chylomicron will have specific proteins on top of it, such as an ApoA and an ApoB48, okay, that basically help tell this guy where to go, okay? Again, we'll talk about the chylomicron a bit more later, but now you can see that the chylomicron is actually going to bud off of the endoplasmic reticulum, perhaps go to the Golgi apparatus, get packaged a bit more, and then go to something known as the lacteal, which is just a specialized lymphatic system, okay? Because if you guys remember from the first ever video in this playlist, I mentioned that fats have to go into something called the lacteal, which is the lymphatic system. Then they end up inside of the thoracic duct, which is a venous circulation, which is in the blood. Okay, so first they go to the lymph, then they go into the blood, and then they start their journey. Okay, and that's going to be in the next video. Okay, we'll talk about it in the next video. And I know I went into a lot of detail here, but it's because I wanted to cover pretty much everything that both advanced students and students that are just taking an introductory level course need to know. So if you don't need to know a lot of this, then you can just dump it out of your head. And if you do, then I'm glad. One last thing I really quickly want to mention is that certain drugs have been developed to actually help with, for example, fat loss or controlling cholesterol levels, okay? And one of them is a drug that inhibits specifically gastric lipase, and another one which inhibits pancreatic lipase. It's actually the same drug. It's called orlistat. Orlistat inhibits both the pancreatic lipase and the gastric lipase, not the lingual lipase though. Okay, so that's orlistat, and it's been proven to reduce fat absorption by at least 30%. Okay, and then another drug that helps with cholesterol is called ezetimibe, and this transporter right here, okay, is the target of ezetimibe. So here is ezetimibe, and it actually targets this NPC 101, which blocks cholesterol absorption to kind of help with controlling cholesterol. But again, with cholesterol, it's important to understand that we actually make more cholesterol in our body, and remember that the free cholesterol from the bile also has to get absorbed. And if that's not absorbed, then what ends up happening to it is it'll get excreted out in the feces, okay? Or some of it might also get reabsorbed at the distal ileum into the biliary system. But again, it's important to realize we actually make more bile ourselves than we ingest not bile. We actually make more cholesterol ourselves than we actually ingest. So these drugs have only limited success. And for example, this actual drug causes a significant reduction of about 18% in blood cholesterol levels. But for patients who can't actually handle that, we'll just say that they should just ingest phytosterols, which cause a reduction of blood cholesterol concentration by 10%. Okay, but that's pretty much going to cover everything with lipid digestion and absorption. If you guys liked the video, please like, comment, subscribe. If you have any questions, comment. I'll be more than happy to answer. And I'll see you guys in the next video when we talk about chylomicron, or actually lipoprotein metabolism.